So this video is going to look at limiting factors of photosynthesis. And we could well ask, well, what is a limiting factor? And a limiting factor is a quantity in a system whose change in amount will noticeably affect the output of that system. So, for example, the rate of photosynthesis is dependent by light intensity, carbon dioxide, temperature, and availability of water. And we might see how light intensity affects the rate of photosynthesis. This axis on the y-axis is rate of photosynthesis. And we can see that if we keep temperature constant and vary, light intensity changes and level of carbon dioxide stays the same, the rate of photosynthesis increases and then levels off. When rate is increasing, light is not the limiting factor. Once it flattens off, light is the limiting factor. Notice we can increase the rate by increasing temperature. We could also increase the rate by increasing the concentration of carbon dioxide. Or we could further increase the temperature. All of these will increase the rate. But initially, when light is the limiting factor, the other variables don't change that much. The other variables don't change the rate that much. But once light is limiting, then the other variables greatly affect the rate because they become the limiting factor. So the point here is there's only one limiting factor at a time. And the things we're going to look at here are discuss the concept of limiting factors in photosynthesis. And related to this, we're going to design or investigate the effect of limiting factors on the rate of photosynthesis. We're going to discuss how to measure the rate of photosynthesis. And from this we can design a P&D lab for you to do. And then discuss how knowledge of these limiting factors can be applied to improvement of plant productivity. So light intensity and carbon dioxide concentration, greenhouse technology. Of course, if you have a greenhouse, which is a house made of glass, we can manipulate the concentration of carbon dioxide or temperature inside of that house. We could also mention hydroponics and growth chambers. So we can move on to those as well. So first of all, what is a limiting factor? It's a variable of a system that causes a noticeable change in output when its quantity changes. So we can look at this again. Light intensity is increasing. The other variables are being held constant. Light intensity increases. Rate of photosynthesis increases and then flattens out. If we raise the temperature at the same concentration of CO2, there's not much change in the rate until the thing becomes light saturated, and then the difference is a bit larger. Another thing we could vary is the concentration of CO2. And notice initially there's not much change in the rate of photosynthesis. I suppose we could mention it flattens out later on. In other words, will become light saturated at a later point in time. And at a higher level of CO2, or a higher temperature, it will flatten out or become light saturated even later. Um, so photosynthesis is affected by three environmental conditions: temperature, light intensity, concentration. At any point in time, only one condition will be limiting. So that's important. Only one factor will be limiting at a time. You can't have two limiting factors. One factor, either temperature, light intensity, or concentration of CO2, will be the limiting factor. Increasing the amount of this substance will increase rate of photosynthesis, but increasing the amount of the other two will have no effect um, until the thing becomes light saturated, of course, and then it has a big effect. Water can also limit the rate of photosynthesis, because if water is in limited supply, the leaf will wilt and stomata will close, and therefore the leaf can't obtain carbon dioxide. And because it can't obtain carbon dioxide, it can't photosynthesize. So this is how light intensity affects rate of photosynthesis. Uh, it increases and then becomes flattens out. At this point, it is light saturated. Low light conditions decrease rate, increasing light intensity. Past the optimum light intensity has no effect. So eventually, light has no effect. Increasing or decreasing optimum temperature results in lower rates of photosynthesis. And this, of course, because photosynthesis is also dependent on enzymes. And then concentration of carbon dioxide. This graph looks a bit like the one for light intensity. So in the first graph, light is the limiting factor. As light is the limiting factor, the rate of photosynthesis increases until a critical value when it no longer has an effect. And beyond this point, light intensity is no longer limiting. Okay, and you can explain this by light intensity determines rate at which ATP and ATP are produced. If they're produced fast enough, they become in excess and can no longer affect the rate of photosynthesis. So eventually, you'll have more ATP and NADPH than you have ribulose phosphate, and therefore increasing the amount of ATP and NADPH even more won't really affect how much carbon dioxide can be fixed and then reduced into phosphoglyceraldehyde. In the case of CO2, if CO2 is in low concentration, then it combines with RUBP as soon as it enters a chloroplast. Hence, rate of photosynthesis increases with CO2 concentration. Eventually, however, concentration of RUBP will limit photosynthesis because you have more CO2 than you have RUBP. So increasing CO2 won't increase the rate again because there isn't enough RUBP around. And temperature affects rate of photosynthesis because of the presence of enzymes. And this again shows the effect of light intensity on photosynthesis. We've discussed this graph already. This is increasing temperature. And notice this is a typical enzyme curve. So once the maximum rate of photosynthesis is reached, this is the optimum level, and it falls off very quickly after that. The breaking of bonds in enzyme structure leads to a change in the shape of that site, um, and increasing molecular collisions due to increased kinetic energy on this side. So temperature also increases the rate of photosynthesis because there's an increase in the rate of diffusion of carbon dioxide, and that's important. One factor reason why temperature increases the rate of photosynthesis is because carbon dioxide diffuses in at a faster rate, so have a greater rate of gaseous exchange. Now, photosynthesis can be measured this way, um, where we have a lamp, we have some photosynthesizing plants in some water. Since they're photosynthesizing, they're producing oxygen, the little bubbles. These will go into this gas syringe and push the cylinder back, and you can make readings at different points in time, and you'll get a graph, and you can determine the rate at any point in time by drawing a 10 tangent to the curve. So it's fastest at the start and slowly begins to slow down as light intensity, well, this is with time in fact. Um, uh, at this point in time, there's no reaction occurring. Um, Really, if it was photosynthesizing all the time, you'd expect it to go at a continuous uh, rate. So this graph, in fact, looks more like a reaction of CO2 with acid. Um, if you have a continuous light being applied to a plant, then you expect the curve to continue as a straight line. So you have a lamp shining on pondweed, and you can uh, alter the rate of photosynthesis by moving the lamp further back from the source. And the further back it is, the slower the rate of photosynthesis. Uh, so how can we increase plant productivity? That is, the amount of carbohydrate in a given period of time can be increased by planting in a greenhouse made of glass or plastic and controlling internal temperature, level of carbon dioxide, and provide lighting. So you can control light intensity and so on using uh, a greenhouse, and therefore you can get more crops being produced. You can also use growth chambers. This is an example of a growth chamber where you have artificial lighting, uh, which you can regulate plant growth. You can regulate, since it's a very small room, you can regulate temperature and levels of carbon dioxide fairly easily. So what is a plant growth chamber? Allow researchers to control environmental conditions when studying plants. Research biologists and other plant professionals can control humidity, temperature, light, and other factors. And growth chambers help plant pathologists fight diseases, and geneticists develop sturdier food crops. They provide essential data for seed germination that the next Norman Borlaug will use to fight off future food crises. And you can read about Norman Borlaug in your own time. But how do plant growth chambers achieve this? Let's look at the agricultural benefit research. Uh, Reproducibility is an essential element of experimental science. And by having some growing things in the growth chamber, we can control the atmospheric conditions very precisely. As studies reproducibility is the ability of other researchers to duplicate the experiments. This allows others to either confirm or falsify the original study. So growth chambers allow this to happen. Growth chambers do this by allowing researchers to set certain variables during their test. Without growth chambers, there will be variation in other factors that are outside of our team's control. Small factors such as addition of a few moments of sunlight a day or air composition can drastically cause dramatic variations in results. And environmental growth chambers ensure equal factors during the whole verification process. So, ideal tests for developing countries. And one of the largest concerns for agricultural scientists is the lack of farmable land. And many countries will face shortages in farmland in the near future. This problem, along with a lack of skilled labour, climate change, and a booming worldwide population, might lead to impending food crises. So in the UK, it is expected there will be a food shortage of farms by 2030, which is not too far away. In Africa and other parts of the world, the problem is not a lack of land, but a lack of arable land. And experts believe that the world's population will grow to 11.2 billion by the mid-2030s. And population increases will lead to a shortage of resources, in particular food. And this will probably leave us with two options. One, people could stop reproducing. Um, as the thing said, this has yet to happen in humanity's history. So it's safe to assume that people are not going to stop having kids. Um, well, I suppose you could look at China, which forced people to have one child per couple for a good period of years. And that was fairly successful. So, and of course, if you just have one child per couple, then the population will fall fairly drastically. We can make a better use of the land we have. That's another one. And growth chamber allows researchers to develop high green products that use less land and produce more food. This also helps determine what regions of the world produce the best of a certain crop. So, growth chambers can help researchers develop high green products. In the future, it is almost guaranteed that food production will take on a global approach to meet the needs of humanity, and certain nations will grow food for the sole purpose of exportation while importing others. We have seen the beginning of this approach over the past 50 years, and this trend will continue. So some nations will produce food which they export and then import from other nations. And I suppose the world could do that, where some countries produce certain food and other countries produce others, and it can all interchange. Although cooperation between countries is probably a bit optimistic. Growth chambers allow research in the United States to test crops in climates like Asia and Africa. That's also possible. I suppose one of the problems here, of course, is that people in Asia and Africa could also do research on crops. The probability is they don't have the financial resources. So perhaps another alternative would be to provide financial backing to people in Asia and Africa so they can test the crops themselves rather than being dependent on the United States. Um, I suppose the US does it so that it can continue to get funding for such research, and therefore their universities will flourish, when perhaps the best bet would be to simply provide Asian and African countries with the financial uh, support to do the research themselves. And then you wouldn't need the growth team. For the research is currently our best hope of staving off food crisis that could lead to millions, if not billions, of people starving to death. This final statement is probably not correct. The best hope, as I said, would be to provide financial assistance to researchers in Asia and Africa so they can do the research themselves. Well, then, a growth chamber helps you make and test predictions. And the hallmark of a good theory is the ability to make predictions about future events. In the case of agricultural science, this occurs by predicting such things as yield and survivability. In the past, a scientist would develop a new type of grain that they believed would survive a shorter growing season. They would then have to test the crop in the real world. If the grain did not survive, it would meant a wasted work and a hungry mouth to feed. And now scientists can make predictions and use growth chambers to simulate a shorter growing season. If the prediction does not hold true, they can immediately begin work on a new solution. So, in other words, it makes things faster. This type of dynamic research leads to the creation of new crops. Researchers can adapt to changing climatic patterns and other conditions. Water shortages could have devastated communities in the past. Now we're able to plan ahead through the use of growth chambers and variability they afford. And that's important. As I say, climates are changing and we need to adapt to uh, kind of periods of drought followed by periods where you have extreme rainfall. And we need to develop plants which are tolerant of these. And that basically seems to finish the presentation. Um, so if we look at this, we looked at light intensity, carbon dioxide concentration. And I said light intensity increases the production of ATP and NADPH. And I suppose the key thing there is that eventually there'll be more ATP and NADPH than there is phosphoglycerate. And therefore, having more ATP and NADPH won't help because phosphoglycerate is being produced at a maximum rate. I suppose one thing there is if suddenly the light was cut off, therefore, the phosphoglycerate would build up, but the phosphoglyceraldehyde would decrease because all the ATP and NADPH would be used up. So, and carbon dioxide. Well, carbon dioxide combines with ribulose bisphosphate. So we'll discuss that. Oh, greenhouse technology. We mentioned that control environmental conditions. Wilting. If a plant wilts, it cannot photosynthesize because the stomata are closed. Hydroponics. We didn't mention that. Hydroponics are simply growing crops without soil. So you just have say a plant growing in a tube. I suppose I could add to that um, later on, but that's clear enough. And this more or less ends today's presentation.